using mouse models to study LCHADD chorioretinopathy and other FAODs. Shannon Babcock received her Bachelor's of Science in Biochemistry from Gonzaga University in 2017 and has recently earned her PhD in Molecular and Medical Genetics from Oregon Health and Science University in Portland, Oregon. While at OHSU, Shannon studied the molecular mechanisms involved with and potential treatments for LCHADD chorioretinopathy using a novel LCHADD mouse model. Yay! Since graduating in May 2024, Shannon has begun a laboratory genetics and genomics fellowship here at the Henry Ford Hospital in Detroit, and where she's working to become a clinical lab geneticist. In her free time, Shannon enjoys playing tennis and spending time outdoors with her dog. And so, Dr. Babcock, on behalf of our community, thank you so much for being here and being willing to present this information. So, please welcome. Thank you.
Thank you for the introduction, and thank you everyone for the invitation to be able to speak today. I think this talk might be a little bit different than what's been presented earlier in this conference, and it's going to focus mainly on how do you study fatty acid oxidation disorders in a lab, so a little bit more at the molecular and cellular level. And one way to do this is by using mouse models, and so we're going to talk about the importance of them as well as benefits that come with using mouse models to study these disorders. I'm going to weave in a little bit of my thesis work that I just completed, which studied LCHADD chorioretinopathy using a mouse model, and I'm hoping this can serve as like an example of how you can find an appropriate model and then how you can use that model. So as most of you probably know, fatty acid oxidation is an important metabolic process that is crucial for energy production. And fatty acid oxidation disorders are a family of these disorders that result from a loss or reduction of function in any part of this pathway. So as you can see on the right-hand side, there's 11 known fatty acid or reported fatty acid oxidation disorders. And these can result, this family includes disorders that result in any part of this pathway. So it could be in the transport of fatty acids into the mitochondria. These are like your CPT disorders. There can be deficiencies in long-chain fatty acid oxidation, such as LCHADD, and TFPD, and then also in deficiencies in medium-chain fatty acid oxidation, such as MCAD. I want to say a couple more things. All of these fatty acid oxidation disorders are rare. They're also monogenic disorders, so they are caused by pathogenic variants in a specific gene. They are also autosomally recessive inherited, meaning that you have to have 2 copies of these genes. And then something that will be important later on is that these disorders result in acylcarnitine profile alterations. And so if you can see on the graph in the 4th column, each disorder kind of has its own unique acylcarnitine profile, and this is important because they serve as biochemical markers for each disorder and can be used for diagnosis. Now, there are many ways that you can study fatty acid oxidations as well as other disorders. One is by doing clinical research, so this is observing patients in specific situations. And while this is very beneficial, there are some limitations. One is that, especially for rare disorders like fatty acid oxidation disorders, you're limited by the number of patients, and this can limit the amount of research that you can do. Another one is that there's only so much that you can ethically study with patients. And so if you want to kind of understand the disorders a little bit more, especially at a cellular or molecular level, you need to kind of turn to model organisms or model systems. One of those is a cell culture model. So this grows specific cells in a petri dish, and we won't talk about that too much today, but the one that we'll talk about is using laboratory animal models. And this is a really big field, or big kind of field. Yeah, there's a lot of different models that you can use. There's fruit flies, zebrafish, rodent models, and then also all the way up to non-human primates. And so we're going to specifically focus on mouse models. And mouse models and rodents models, they make up about 95% of all laboratory animals models used. And there's a lot of benefits to using mouse models. One is that they're very well understood and studied. Another one is that they have a short reproductive cycle and lifespan. So this allows you to conduct research at a little bit faster pace. They're also easier to maintain than other animal models, so it makes the research a little bit cheaper as well. They are also genomic and physiological similarities between mice and humans, and this is really important when studying metabolic disorders and fatty acid oxidation, because metabolism is a connection of many, many different pathways, and they're influenced by your diet and also the environment. And so by using a mouse, you're able to kind of mimic that that complexity that occurs in human biology, and see how this disorder is affecting kind of the whole system. And then finally, especially when studying genetic disorders like FAODs, mice with the invention of CRISPR-Cas9 and other gene editing technologies, you can easily manipulate mice to have specific variants and affect specific genes. And we'll kind of mention this a little bit later on too. One really big benefit of using mouse models is that you can study molecular mechanisms. And the reason that you'd want to study molecular mechanisms is because this kind of looks at what is exactly happening in the cell for a cell to become dysfunctional or cause them to die, because it's really this dysfunction and death that leads to the presentation of specific symptoms. And so by looking at the molecular mechanisms, you're really trying to identify what is happening inside of the cell, and this can lead to much better and targeted treatments for patients. Another thing that this mouse models are really beneficial for are being used to study and evaluate potential treatments. So even some clinical trials do require you to have some sort of testing before you can take a potential treatment to clinical trials. You need to do it in an animal model, and so this is really beneficial for those too. Now, as you can see on the graph on the left, there are fatty acid oxidation disorders have really been, or mouse models have been really beneficial in fatty acid oxidation disorder research. So the majority of fatty acid oxidation disorders that have been reported do have one or more mouse models that have been created to study these. Some of these models have been used to study molecular mechanisms involved in phenotypes such as your cardiomyopathy, exercise intolerance, and then also why do patients become hypoglycemic during fasting. They've also been used to evaluate potential treatments. So they've been used to evaluate things such as the effect of MCT and triheptanone, if you are familiar with those. So overall, they've been really important. So in order to be able to use a model to study a specific disorder, you want to make sure that it's an appropriate mouse model. So there's a couple of things that you can look for when determining this. One is you can look to see if it's genetically similar, especially if you're studying a genetic disorder. So you want to make sure that the mouse and the human have pathogenic variants in a similar gene. So for example, if you wanted to study MCAD, patients have typically pathogenic variants in the gene ACAD-M. You would want to find a mouse model that also has pathogenic variants in the ACAD-M gene in the mouse. Another thing you'd want to look for is it biochemically similar? So does it have the same block in fatty acid oxidation as patients do? So for example, if you want to study VLCAD, this is a long-chain fatty acid oxidation disorder, you would want to find a mouse that also has a block in long-chain fatty acid oxidation and not something that has a block in like medium-chain fatty acid oxidation, because they are different. One way that you can test this is to see if they have similar acylcarnitine profiles, which I mentioned before are biochemical markers of these disorders. And so you can see if the mouse has a similar block by looking at the acylcarnitine profile and finding one that is similar to patients. And then finally, and probably one of the more important ones, is it phenotypically similar? Does it actually develop the same symptoms as the disorder you are trying to study? And does it kind of progress in the same way as well? So from here on out, I'm going to try to tie in some of my research that I did that studied LCHADD chorioretinopathy using a novel LCHADD mouse model. And so I'll explain what all of that means here in a second. But I hope this kind of can serve as an example of how we were able to find a mouse that was an appropriate model, and then the benefits that we were able to, or the benefits of using the mouse model and what we were able to find using this mouse. So LCHADD stands for long-chain 3-hydroxyacyl-CoA dehydrogenase deficiency, and this is a long-chain fatty acid oxidation disorder. Patients can't metabolize fatty acids that are between 12 and 20 carbons in length, and one that is probably most common is palmitate, which is 16 carbons in length. LCHADD patients commonly have a pathogenic variant, this G1528C missense variant in the HADHA gene. And these patients, again, are homozygous for it, so they have typically two copies of this gene. And what this variant does is that it knocks out specifically the LCHADD enzymatic activity. And this is the third step of long-chain fatty acid oxidation. And what results from this is that you have a unique accumulation of partially metabolized intermediates, such as the 3-hydroxyacyl-carnitines, which will be important later on in this talk as well. So patients develop a variety of symptoms. Some of them are common among all fatty acid oxidation disorders. This includes cardiomyopathy, rhabdomyolysis, and metabolic crises. But what makes LCHADD unique is that patients develop very specific symptoms seen only really in LCHADD patients. So one of those is peripheral neuropathy, and the other one is chorioretinopathy, where patients will lose their vision at a pretty young age. And again, this is only seen in LCHADD patients, and this is the one that we're focused on, I was focused on in my PhD in studying, and the reason why is because it's currently not preventable. It's currently not preventable using the current dietary management. So we needed to find, the goal was to find a new potential treatment for LCHADD chorioretinopathy. The problem with LCHADD research is that it has been very limited because there hasn't been a mouse model that mirrors LCHADD. So specifically, there hasn't been a mouse model that has that G1528C missense variant. There hasn't been a mouse that accumulates those hydroxyacyl-carnitines, nor has there been a mouse that really develops the chorioretinopathy that you see in LCHADD patients. And so for us to be able to study LCHADD chorioretinopathy with a mouse, we needed to generate one. And so the first step was to make sure that this mouse was genetically similar to patients. And again, patients have that G1528C missense variant in the HADHA gene. And so the Gillingham lab used CRISPR-Cas9 to insert that specific variant into a mouse line. And so now that this mouse is homozygous, so it's two copies of that G1528C variant in HADHA, making it genetically similar to patients. We then wanted to make sure that this mouse was biochemically similar to patients. And specifically, we needed to make sure that this mouse had decreased long-chain fatty acid oxidation and also had a block in that LCHADD enzymatic activity, which resulted in those accumulation of hydroxyacyl-carnitines. So I'll first start off with how we figured out that it has decreased long-chain fatty acid oxidation, and we used a palmitate oxidation assay to do this. So this is an in vitro assay where you're able to take some tissues. We specifically looked at the heart and the liver to see how much of a radio-labeled palmitate these tissues were able to actually metabolize and break down. And so as you can see from the graph on the far right, in both males and in females, the LCHADD mice, which are in orange, are unable to metabolize as much of that radio-labeled palmitate as the wild type, which are our healthy, normal mice that we had, indicating that, yes, the LCHADD mice do have decreased long-chain fatty acid oxidation. We then wanted to make sure that this was due to a block at specifically that LCHADD enzymatic activity step. And you can see, and by doing this, we looked specifically at the acylcarnitine profiles, looking to see if these mice accumulated hydroxyacyl-carnitines. And you can see over on the graph on the left, or the two graphs on the left, that indicated by the red boxes too, that the LCHADD mice, again in the orange, have higher levels of those hydroxyacyl-carnitines in the plasma or in blood, indicating that, yes, overall, this mouse does have decreased long-chain fatty acid oxidation, as well as decreased LCHADD enzymatic activity, making it biochemically similar to patients. So now that we knew that the mouse was genetically and biochemically similar, we needed to make sure that this mouse was phenotypically similar. So specifically because we wanted to use this mouse to study the chorioretinopathy, we needed to make sure that this mouse lost its vision and developed chorioretinopathy. And so just a quick background about this, LCHADD chorioretinopathy means that patients lose their vision at a pretty young age due to the atrophy of the choroid and the retina. And both of these are located in the very back of your eye. The choroid is a blood vessel that vascularizes the retina, so it's important for transporting nutrients to the retina. And the retina is a bunch of different cell layers that are important for responding to light, initiating an electrical signal, and then sending that to the brain where it's interpreted as your vision. Now, again, the retina is made up of several different cell types. And we're going to focus mainly on the outer retinal layers, specifically the photoreceptors and then the retinal pigment epithelium. The photoreceptors may be a little bit more well known. These are your rods and your cones, and these are important for responding to light and then initiating that electrical signal to the brain. The retinal pigment epithelium, or the RPE, is probably a little bit less known. But this is a really important cell layer that maintains the function of the photoreceptors as well as the choroid. So if you do end up losing your retinal pigment epithelium cells, this can cause photoreceptors to die and eventually the vision loss. So from clinical studies, we were able to characterize LCHADD chorioretinopathy, and it's from these studies that suggest that it's the retinal pigment epithelium cells are the first cell layer that begins to die and is really what initiates the chorioretinopathy. So what you're seeing on the 4 pictures on the right, these are fundus images. And if you've ever been to an optometrist, you've probably had this done. They are just photos of the back of your eye, specifically the central vision. So you can see, I don't have a pointer, I don't think, but the big white spot, that's your optic nerve, and then a little bit dark circle, that's your macula. And in a normal fundus image, you want to see an eye that is uniformly pigmented, which would be located number 1 picture. It's a nice uniform pigmented color. LCHADD patients, the first sign of LCHADD chorioretinopathy in patients is that fundus images begin to have this like mottled appearance, and you start to begin to see these areas of hypopigmentation, as seen in the picture 2A. This is what is indicative of RPE degeneration and why we believe that the RPE is the first layers to degenerate. And so as the chorioretinopathy progresses, seen in 3A and 4, you start to lose more and more pigmentation, which suggests more loss of the RPE, and then you also see photoreceptor and choroid degeneration, which is really what ultimately leads to the loss of the vision. So from the clinical studies, we wanted to make sure that our mouse model had a chorioretinopathy that was similar to patients, specifically that they lost their vision over time and that it was due to increased RPE degeneration. And so we first looked at visual performance of the mice. So how well are LCHADD mice able to see over time? So we took these visual performance measurements at 3, 6, 10, and 12 months of age. And the test that we use to do this is called optokinetic tracking, or OKT. And how this works is that a mouse is placed on a pedestal inside of the box, and on the sides of the boxes are these different gratings. It's the frequency of those gratings that determine the visual performance. So as you increase the frequency of that grating, that means you have better visual performance. And so the test works, you keep altering these frequencies to find the highest frequency that the mouse can see. And the way that you can tell a mouse can see it is that the mouse's head will move in the direction of the grating. So the grating can move clockwise or counterclockwise, depending on if you want to measure the left eye or the right eye. And you can tell the mouse can see it because its head will also move in the direction of the grating. So it will move clockwise with the grating, or it will move counterclockwise. And so if you look at the data on the right, that we took from the OKTs, you can see that especially starting at 6 months of age, these LCHADD mice have decreased visual performance. Again, those are the ones in the orange, compared to the wild type, which are the blue, suggesting that at 6 months, these mice have decreased visual performance. But this vision continues to decrease over time. So if you look at the percent reduction from 3 to 12 months, the LCHADD mice have a reduction of about 30% in visual performance from 3 to 12 months, whereas wild-type mice have about 14% reduction, indicating that these mice are losing their vision at a faster rate than the wild type, and you can see a pretty significant difference starting around 6 months of age. We then wanted to see if these mice had increased RPE degeneration. And to do this, we calculated the percentage of eyes in each group that had those areas of hypopigmentation. So I don't know if you can see it all that well, but the images, especially on the bottom under LCHADD, the black arrows are pointing to areas of those hypopigmentation, which are indicating RPE degeneration. And so we calculated the percentage of those eyes in each group at the same time points, those 3, 6, 10, and 12 months of age. And you can see that, again, at 6 months of age, the LCHADD eyes, about 30% of them had some area of hypopigmentation, indicating RPE degeneration, whereas only 10% of the wild type did at that same age, indicating that, yes, LCHADD mice have increased RPE degeneration. And this also continues to decrease, or I guess the degeneration continues to increase over time, where it gets up to about 60% at 12 months of age. And so all of these kind of together just say that, yes, this mouse is a good model to study LCHADD chorioretinopathy. It is genetically similar. It's homozygous for that G1528C variant. It's biochemically similar, so it has an increase of hydroxyacylcarnitines and decreased long-chain fatty acid oxidation. And also now we show it's phenotypically similar. It develops a chorioretinopathy that is similar to what we see in patients. And so now that we know that this is a good mouse model to study, LCHADD chorioretinopathy, we can use it to study molecular mechanisms involved in it. And again, analyzing molecular mechanisms would allow us to identify kind of the root causes of what is happening inside specifically those RPE cells, in our case, and to be able to potentially develop more targeted or better therapies for patients. Mouse models are really important for doing this because they allow you to do experiments that you couldn't really do in patients. And I'll explain here in what we were looking for in the mouse model. So we know that LCHADD chorioretinopathy is initiated by the loss of those retinal pigment epithelium cells, but we don't know what is causing those RPE cells to die. And so there are a couple of hypotheses out there, and one that I'll mention today is that those hydroxyacylcarnitines and other partially metabolized intermediates are actually toxic to the RPE cells and causing them to die. And it's been shown that the RPE actually do rely on fatty acid oxidation, not only to make energy for themselves, but also to maintain kind of the photoreceptors as well. And so the hypothesis is that that the RPE is creating these hydroxyacylcarnitines and other toxic intermediates, and it's accumulating inside of those cells and then causing those RPE cells to die, which initiates that chorioretinopathy. Now, in order to test this, one thing you could do was somehow be able to correlate the hydroxyacylcarnitine concentrations inside of the retinal pigment epithelium cells and then correlate it with vision performance. But especially if you wanted to do this in clinical studies, you really couldn't, because you can't measure acylcarnitine profiles in RPE, because that would mean you'd have to, take out the RPE, and that itself would cause vision loss, right? So this is where mouse models come in handy, is where you can actually do these types of experiments. And so we were interested in looking at that during my PhD. And so we isolated RPE from 15-month wild type and LCHADD mice, and we looked at acylcarnitine profiles in them. And what you see is that in the LCHADD RPE, there is an accumulation of those long-chain hydroxyacylcarnitines, as you can see by the two, or the stars at the top. Those are the hydroxyacylcarnitines. And so when you pair this with the data that I showed previously, where these mice also have decreased visual performance, you can then start to develop an association between hydroxyacylcarnitines and visual performance. Now, I want to make sure that this is not enough to confirm this hypothesis. There's many, many more studies that need to be done. But I wanted to kind of just include this to show that you can then, this is one benefit of using a mouse model, is that you can actually correlate and start to develop or start to confirm this relationship where if you potentially have increased hydroxyacylcarnitines in the RPE specifically, you can have decreased vision loss. And so this is something that you can do or kind of iron out a little bit more going forward. Oh, that was the wrong way, sorry. Okay, so the last section that I'll talk about here is not only are mouse models important for studying molecular mechanisms, but they're also important for studying potential treatments. Mouse models, specifically fatty acid oxidation disorders, again, have been used to study MCT and also triheptanoin, but it also allows you, using mouse models, you can find a little bit more, advanced, I guess, treatments, ones that maybe aren't currently being used right now. And one treatment that we were interested in was gene addition. And gene addition has been a really important or really sought after treatment for other retinopathies. If you've ever heard of Luxturna, that was the first FDA-approved gene addition treatment. So that was used to treat patients who lost vision for a specific reason. But it's also been used and studied in other fatty acid oxidation disorder mouse models. So this includes, it's been used to study VLCHADD and also MCAD mice. And the way that gene addition works is that it aims to treat a disease by adding a functional copy of a gene to specific cells to treat and prevent the disorder. So here's my little schematic. Fatty acid oxidation disorder patients currently have a gene that has pathogenic variants in it, and those variants cause a specific protein or it cause for the encoding of a deficient protein. Gene addition works by hopefully adding a functional gene to those cells, and so not only then would that cell produce the deficient protein, but it would then produce a functional protein, and this functional protein would be able to restore the enzymatic activity that patients were deficient of and potentially prevent the disease from occurring. And so this was one that we were very interested in. We wanted to test this in our mice to see if it could be a potential treatment. And so we injected 2-month wild type and LCHADD mice with a functional copy of HADHA gene, specifically targeting the RPE cells. And then at 12 months, we looked to see, did it prevent the mice from losing their vision? And did it prevent RPE degeneration? And then also, was it able to restore fatty acid oxidation in those cells? So we'll first look at the gene addition, or the visual performance results. And we saw that this gene addition, I do want to say that this study, the sample size was really small. So many, many more studies will need to be done in order to confirm this. But it was really exciting preliminary results. So in the visual performance, at the 12 months, which again is the graph on the left, you can see that mice treated with the gene addition, which are, in this case, looks like LCHADD, or the virus ones, those mean gene addition. But the LCHADD treated with gene addition had retained their visual performance to the level of the wild type, whereas untreated LCHADD eyes had decreased visual performance, suggesting that gene addition was able to preserve some of the vision in these mice. We also looked to see if it prevented RPE degeneration, where again, an image of that RPE degeneration is there in the middle with the white arrow. And we calculated the percentage of eyes in each of the groups to see how much of those eyes had the hypopigmentation. And you can see that LCHADD eyes that were not treated with gene addition, which is, again, the LCHADD PBS, 100% of those eyes developed some hypopigmentation indicating RPE degeneration. Whereas the LCHADD eyes treated with the gene addition, which again is the very bottom, labeled as LCHADD virus, about 67% of those had RPE degeneration. And so this suggests that it was able to prevent at least some RPE degeneration. It wasn't able to prevent all of it at this stage. As you can see, both the wild type mice had 25%, so it wasn't able to completely prevent it, but it did have lower RPE degeneration when compared to untreated eyes. And then I wanted to show kind of another really exciting result that we had. So we wanted to see if the gene addition was able to restore fatty acid oxidation to RPE cells. And so we looked at the acylcarnitine profiles in order to do this. So as you can see, the LCHADDs treated with the gene addition, which are the orange bars with the stripes, they had lower levels of hydroxyacylcarnitines, which is the very far, the C18OH and also the C16OH. They had less levels of hydroxyacylcarnitines when compared to untreated LCHADD-Is, which again are just the solid orange bars, suggesting that LCHADD enzymatic activity was restored and also the long-chain fatty acid oxidation was also restored. Now again, it wasn't restored all the way to wild type. It didn't completely fix all of the cells as they were still higher than the wild type ones, but it showed partial recovery, which was exciting. And if you if you associate this with what we saw, it also builds another or provides another evidence that there may be an association between hydroxyacylcarnitines and visual performance, where if you're able to lower hydroxyacylcarnitines, specifically in the RPE, you would be able to preserve some vision. And so again, that is just our preliminary evidence. Much more will need to be done in order to confirm that. But that was an exciting piece and could potentially lead to potential treatments. So just to wrap this up, I hope I conveyed that mouse models are really important in studying disorders, specifically fatty acid oxidation disorders. They are crucial in studying molecular mechanisms because it allows you to do experiments that you wouldn't necessarily be able to do in clinical studies. And it provides more information as to what is happening to these disorders and can provide for better treatments. It also allows you a preclinical model to studies where you can test those potential treatments before going to clinical trials, and that is required by most clinical trials anyways. I talked a lot about my PhD work here, where I used a novel LCHADD mouse model to study LCHADD chorioretinopathy. This mouse is a great model. It was genetically similar and biochemically similar to patients and developed a chorioretinopathy that was similar. And so because it's an appropriate model to study LCHADD chorioretinopathy, we were able to investigate specific molecular mechanisms that could be involved in the RPE death that we were seeing. Specifically, maybe hydroxyacylcarnitines and vision are related and correlated. And also we were able to test gene addition as a potential treatment for the chorioretinopathy. And so this mouse model, it is a really big step in furthering research into LCHADD chorioretinopathy and finding a potential treatment. And so with that, I have some acknowledgements. Of course, I would like to thank the Gillingham Lab, which is the lab I did my PhD research in, all the past and present members. I had some collaborators, so Dr. Renee Riles' lab at KCI Institute at OHSU was really crucial in doing a lot of the vision experiments with me. And then Dr. Sander Houghton at the Icahn School of Medicine at Mount Sinai, he was a collaborator, and we actually wrote a fatty acid oxidation disorder mouse model review that has been recently published. Of course, our funding, we can't do research without funding, so specifically Peterson Foundation and held donation for their donations, and then also OHSU, which is the place that I did my research at, specifically the Molecular Medical Genetics Department, for allowing us to do that. And so with that, I will take any questions.
So we're running a little bit behind, so in lieu of questions, if you have questions, she's just agreed on the spot to stay for our expert panel. So hold those questions. We have children in the lobby that are very hungry and need their parents to help them figure out what's appropriate to eat. So please go join your children or yourself and go feed yourself, and we will meet back here at 1 o'clock, and it'll be a perfect timing because Dr. Arnold is going to talk about FAOD, the effects on families and food and relationships.